Assalamualaikum and greeting. My name is Muhammad Farish Daniel bin Shamsul Imran, a final year student of Bachelor of Science honored in Biology. Today I am going to present my final year project with a title, The Impact of Cleaning Methods on Microbiological Loads in Glass and Plastic Food Packaging Jar under the supervision of Dr. Wan Nurhayati binti Wan Hanafi. This is my table of contents. Microbes, or also known as microorganisms, are all over the place, present on the surface, in the air, inside the human body. One of the main causes of microbial contamination in food preservation is unsafe or unhygienic food packaging practices. Different types of food packaging jar have varying effects on microbial contamination, with bacteria tending to attach more to hydrophobic surfaces like plastic compared to hydrophilic surfaces like glass. Despite being newly bought, food packaging jar may or may not be free from microbial contamination. This study aims to determine the level of microbial contamination in food packaging jar and evaluate the effectiveness of different types of cleaning methods by examining the microbiological load after cleaning. There are three objectives of this study. The first was to evaluate the microbiological loads before and after cleaning food packaging jar. The second was to observe the microbiological load between plastic and glass food packaging jar. And lastly, the third was to analyze the effectiveness of cleaning methods to eliminate microbiological loads in food packaging jar. There are two types of food packaging jar prepared, which are glass and plastic. Each food packaging jar was underwent various types of cleaning methods, including cleaning with pipe water, cleaning with water and soap, UV light exposure, and hot temperature exposure. One controlled or unwatched food packaging jar was prepared to check the microbiological loads before cleaning. Each jar was replicated 3 times. After cleaning, each jar was poured with distilled water to make a sample solution. Each solution was diluted 2 times with a ratio of 1 ml of sample solution and 9 ml of distilled water. The diluted sample was then transferred to the media culture using spread plate method. The culture were incubated at 33 to 37 degrees Celsius for 48 hours. The data were observed and compared to their microbiological loads. The first result showed the microbiological load in unwashed food packaging jar. As shown in the picture, there is no presence of microorganism group. This suggests that the food jar were free from microbial contamination. This result acts as the control for other cleaning methods. The second result, which is cleaning with pipe water, and the third result, which is cleaning with soap and water, show there is no sign of microbial contamination or growth in both plastic and glass food packaging jar. Follow the control result. The fourth and the fifth result show that there is the presence of microbial growth in the media culture. As shown in the result above, the microbiological load before and after were the same due to the fact that the food packaging jar was free from microbiological contamination from the beginning. There are no differences between microbiological loads between plastic and glass food packaging jar, as both indicate no presence of microbiological group. The control set served as a baseline, demonstrating that both types of jar, plastic and glass, remain free from contamination. The food packaging was free from the bacteria from the beginning. This suggests that the factory already produced the food packaging jar sterile from microorganism contamination by applying good manufacturing practices (GMP). This was proven by the absence of microbiological loads in each media culture prepared. However, upon UV light and high temperature exposure, microbial loads still detected in the media culture. This suggests that while cleaning methods play a crucial role, certain environmental conditions can still lead to contamination. The observed microbial present under this condition underscore the need for a robust cleaning protocol to mitigate the risk of microbial transfer during handling and storage. Interestingly, the use of a Bunsen burner, typically employed as an aseptic tank, did not guarantee a sterile environment in our experimental setup. In contrast, the laminar flow hood significantly reduced microbial contamination during sampling handling, highlighting its effectiveness as a complementary measure to cleaning methods. This finding emphasized the complex interplay between cleaning methods, environmental conditions, and aseptic tanning in maintaining microbial safety in food packaging. In conclusion, while cleaning methods are essential in reducing microbiological load in glass and plastic food packaging jars, their effect can be altered by environmental factors. Adopting comprehensive strategies that integrate effective cleaning practices with appropriate environmental control is critical for ensuring food safety and quality throughout the packaging processes. However, note that the newly bought food packaging jar still need cleaning to ensure it is completely clean and safe to be used. For future research, it is recommended to study the effect of different types of food packaging jars on dry and wet food to enhance food shelf life. These are the list of references that I use. I think that's all from me. Thank you.